So we will discuss about now carbohydrate handling. Okay, carbohydrate are the main energy source of most diet. Okay, carbohydrate is the most energy source. Okay, they provides 17 kilojoule or 4 kilocalorie of energy per gram. Carbohydrate is digested in the mouth and small intestine. Okay, it's the most main energy source of most of diet. It provides 17 kilojoule or 4 kilocalorie of energy per gram. That's the carbohydrate. Okay, carbohydrate is digested in the mouth and in the small intestine. Okay, in the mouth and in the small intestine. Dietary carbohydrate. Most dietary carbohydrate is in the form of polysaccharide. The principal ingested polysaccharides are starch, which is derived from the plant sources, and glycogen, which is derived from the animal sources. Okay, starch is derived from the plant sources, and the glycogen is derived from the animal sources. Other sources of carbohydrate are monosaccharide, that's the glucose, fructose, and galactose, and disaccharide. That's the maltose, sucrose, and the lactose. Okay, monosaccharide is the glucose, fructose, and galactose, and the monosaccharides are the maltose, maltose, sucrose, and the lactose. Okay, dietary fiber consists of indigestible carbohydrate. Dietary fibers uh, are consist of indigestible carbohydrate found in the plant food, such as cellulose, lignin, and the pectin. Although it does not provide energy, fiber adds bulk to the bowel contents and increases the gut motility. Okay, this fiber adds bulk to the bowel content, bulk add bulk to the bowel content and increase the gut motility, and decreases the absorption of toxic compounds due to its binding properties, including the some carcinogens. Okay, dietary fibers consist of indigestible carbohydrate found in the plant foods. Okay, so like cellulose, lignin, and pectin. Okay, although it does not provide energy, fibers add bulk to the bowel content and increases gut motility, and it decreases the absorption of toxic component due to binding properties, include some carcinogen. Then carbohydrate digestion. So how it digests? So polysaccharides are digested by amylases. Okay, polysaccharides are digested by the amylase, produced initially by the salivary gland and later by the pancreas. Okay, to produce maltose, maltotriose, and limit the dextrins with short branches. Okay, these are broken down further by the enzyme released from the intestinal brush border, like maltase, isomaltase, sucrase, and lactase, into the final products of carbohydrate digestion. The monosaccharides, glucose, fructose, and galactose, which can be absorbed by the enterocyte. Okay, polysaccharides are digested by the amylase, produced initially by the salivary gland and then by the pancreas to produce maltorides and limit the dextrins with short branches. These are broken down further by the enzyme released from the intestinal brush border, maltase, isomaltase, sucrase, and lactase, into the final products of the carbohydrate digestion. These are the monosaccharides, glucose, fructose, and the galactose. Okay, polysaccharide to the monosaccharide, which can be absorbed by the enterocyte. Okay, and this monosaccharide will absorbed by the enterocytes. Monosaccharides are transported across the apical membrane of the enterocyte by means of the co-transport molecules, such as the sodium glucose co-transport as CLT1, that link their inward movement with that of sodium down its concentration gradient, and gradient. Okay, the sodium gradient binding maintained by sodium potassium ATPase pump. At the basolateral membrane, monosaccharides leave the cell either by simple or facilitated diffusion down the concentration gradient and enter the circulation via the rich capillary network in the villus. Okay, so the monosaccharide will transport across the apical membrane of the enterocyte by means of the co-transporter molecules such as the sodium glucose co-transporter. Okay, sodium glucose co-transporter that link the inward movement of the with the sodium down in its concentration gradient by the sodium potassium ATPase pump at the basolateral membrane, and monosaccharides leave the cell either by simple or facilitated diffusion down the concentration gradient and enter the circulation via the rich capillary network in the villus. Okay, these are the glucose and the sodium. Okay, so the SCLT1 transporter will uh, do this transport in the enterocyte, okay, from the lumen to the enterocyte, and this will be facilitated by the sodium potassium ATPase, okay, from the capillary, and the glucose is lastly transport into the circulation via the glucose transport. Okay, now cholecystokinin. So cholecystokinin, CCK, is secreted by the eye cells of the duodenal and jejunal mucosa. Cholecystokinin is secreted by the eye cells of the duodenum and the jejunal mucosa. Okay, duodenum and the jejunal mucosa. Cholecystokinin. So, stimulating factors. Okay, cholecystokinin release is stimulated by the presence of small peptides and amino acid in the duodenum. Presence of small peptide and the amino acids in the duodenum, and presence of fatty acids and monoglycerides in the duodenum. Okay, small peptide and the amino acids, fatty acids, and the monoglyceride, okay, in the duodenum. In inhibitory factors, so the release of cholecystokinin is inhibited by the somatostatin. Cholecystokinin release is inhibited by the somatostatin. Okay, function CCK act to increase the production of bile, okay, stimulate release of stored bile via contraction of the gallbladder and the relaxation of the sphincter of Cody. Okay, it causes the production of the bile and the secretion release of the bile. Okay, production and release of the bile by the cholecystokinin is done. Okay, potentiate secreting induced stimulation of the pancreatic bicarb secretion. Okay, potentiate secretin induced stimulation of the pancreatic bicarb secretion, then stimulate the pancreatic enzyme secretion, inhibit gastric emptying, inhibit gastric acid production, and stimulate secretion of pepsinogen from the chief cells. Okay, stimulate the secretion of the pepsinogen from the chief cells. Okay, so gastrin, cholecystokinin, secretin, gastrin inhibitory polypeptide. Okay, this we will see now. So, gastrin, G cell is released from the G cells in the pyloric antrum. Okay, gastrin is secreted by the G cells in the pyloric antrum. Okay, uh, released from the G cells of the pyloric antrum. Okay, this is the stomach and this is the pyloric antrum. Gastrin is secreted by the G cells of the pyloric antrum. The pancreas stimulated by the presence of the small peptides and amino acids in the stomach, and the gastric distension, raised gastric pH, and the vagal stimulation. This will cause the secretion of the gastrin. Inhibited by the low gastric pH, somatostatin secreting cholecystokinin gastric inhibitory polypeptide act to stimulate acid secretion from the gastric parietal cells, stimulate pepsinogen from gastric chief cells, and increase gastric motility, stimulate growth of gastric mucosa. Okay, act to stimulate the acid secretion from the gastric parietal cells, stimulate pepsinogen from the gastric chief cells. Okay, then increase gastric motility and stimulate growth of gastric mucosa. Then cholecystokinin. So cholecystokinin will secreted from the eye cells of the in the duodenum and the jejunum. Okay, eye cells. So the presence of fatty acids, monoglyceride, small peptides, and amino acids will in the duodenum will stimulate the CCK secretion, and the somatostatin will decrease. Okay, so here also the somatostatin will decrease the secretion. Here also the somatostatin. Okay, this will decrease the all the secretions. Okay, uh, then cholecystokinin act to increase the production of the bile, stimulate release of the stored bile, all that we have seen just now. Okay, then secretin. It's secreted by the S cells of the duodenum. In the duodenum, secretin will be from the S cells. A low pH and the presence of fatty acids in the duodenum will stimulate the secretion of the secretin, inhibit gastric acid secretion, inhibit gastric emptying, stimulate pepsinogen secretion, stimulate pancreatic bicarb secretion, increase pancreatic enzyme secretion, and increase the bile production. All the effect as the cholecystokinin and uh, gastric inhibitory polypeptide. Okay, gastric inhibitory polypeptide (GIP). Okay, it's secreted by the K cells in the duodenum and the jejunum, same as this, but from the K cells. Okay, the presence of fatty acid, amino acid, and orally administered glucose in the duodenum. Okay, administered glucose into the duodenum. Inhibit gastric acid secretion and stimulate the insulin release. Okay, because of the glucose. Uh, okay, uh, sorry. Uh, Inhibit the gastric acid secretion, stimulate the insulin release. Okay,